Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science Podcast. I'm Ayesha Rashid, Senior Life Science Journalist at Xtalks.com. And this week we have a very special guest on our show. We have with us today Louis Bender, who is the founder, chairman, and CEO of Intensity Therapeutics, which is a company developing first-in-class immune-based intratumorally injected cancer therapeutics. The company's lead product candidate is currently in human clinical studies for the treatment of various refractory solid tumors. More about Louis, he has almost 3 decades of biopharmaceutical leadership experience, and he's helped take innovative new products from discovery through to product approval. Louis has held various leadership roles at biopharmaceutical companies, including Interleukin Genetics and Emisphere Technologies. During his career, Louis has partnered with several major pharmaceutical and biotech companies, with success raising capital from institutional investors through multiple types of transactions. So to hear more about the work that Louis is leading at Intensity Therapeutics, let's turn to him now. Thank you so much for being on the show today.
Well, Ayesha, thank you for having me. I'm always happy to talk about what we're doing at Intensity.
And we are so happy to have you. Louis, you've been in this space for a very long time, have had a very illustrious career, and are continuing to lead some fantastic work at Intensity that we'll talk a bit more about in just a bit. But I'd like to first start off by asking you about your journey in biopharmaceutical and biotech and what led you to found Intensity Therapeutics.
Well, again, thanks for the opportunity, Ayesha. Look, my career has been not a planned experience. It's sort of a Monte Carlo random walk through life. After I, graduated and I got a master's degree in chemical engineering, an undergraduate degree in chemical engineering. Then I worked for 9 years, actually, in making materials for various applications, including biomedical. Half of the time was living in Switzerland for 5 years, working in that field. And then, missing America, I came back and I went back for an MBA and a Master of International Studies, an MBA at Penn's Wharton School. And that's when I really got involved actively in biotech, in the biotech industry. That's where I started out at Emisphere and head of business development, and really enjoyed. I was there 14 and a half years. And what I learned at Emisphere was the entire drug development process. I learned the toxicology, pharmacology, quality control, clinical development, manufacturing. I was head of manufacturing and process development and business development, pharmaceutics. And then I became president and interim CEO. And I really understood this technology and the need for good delivery as a function of how we can get improved efficacy and improved safety by delivering the drugs where they need to go. You can make a better and safer product. And so after I worked in the genetics field for about 4.5 years, I started to understand cancer a little bit. And I kind of thought of cancer as an engineer as opposed to a biologist. I thought about it as a physical process. You have these growths in the body that you can see, tumors. You've got to find a way to neutralize them. And at the same time, there's lots of cancer that you don't see, especially in metastatic disease. You don't see all the tumors. They could be anywhere in the body. So you have a problem of how do you treat a tumor or multiple tumors, ones you see and ones you don't see. You don't even know if you have them. That was the problem, right? That's what we've been facing. In current therapies, I looked at current therapies. You have systemic chemotherapy, you have systemic immunotherapy, and in systemic chemotherapy, the drug goes everywhere. It poisons the entire body. Very little reaches the tumors. A lot of the tumors don't have blood vessels, especially the bigger ones. They're very erratic, and so chemotherapy, when you get multiple high tumor burden metastatic disease, is very ineffective. Immunotherapy only works in the cancers that can be recognized or the most mutated ones. 80% of the immunotherapies are in melanoma, lung, and renal, and for pancreatic, colon, and metastatic breast cancer, you don't really have a very good outcome for immunotherapy. So you have a problem with systemic and you have a problem with immunotherapies. What can we do? So I was exposed to this delivery technology at Emisphere, and that delivery technology is a molecular transport, and it is actually comprising the drug ribelsis, which I helped to create. I created the scale up, the process development for the molecules that are used to move the oral GLP-1 molecule through the mouth, into the gut, through the gut, into the blood safely without harming the gut and without harming the molecule. So with that knowledge of how to move molecules, I got an idea about 12 and a half, 13 years ago, when immunotherapies were starting to come into play. And I felt that the problem with immunotherapies was that the cancer can't be recognized by the immune system as easily because it comes from your own healthy cells. So what can we do about that? And how do we kill the tumors we don't see and the tumors we see? So I thought, well, if I could convert the tumor, a large bulky tumor, the size maybe of a can of soda, if we could kill that and convert it in a way that's immunologically activating, perhaps we could train the immune system on your patient's own cancer. There's nothing more personal about the cancer as a potential site for vaccination or antigen than your own tumor mutated the way it was, dying in its natural environment in a way to stimulate an immune response. So I went through so I quit my job as a CEO, and I started this company in my basement. Literally did some of the first chemistry. I partnered quickly with the National Cancer Institute, and we started doing screening on this delivery technology to how we can move molecules through tissue. And I identified, after 2 years of work, a formulation that can be injected into the tumors. The idea was always to try to inject into the tumors and kill the tumors. And we identified a very potent formulation comprising 2 proven IV, normally given IV drugs, cisplatin and vinblastine, very toxic, gram for gram, some of the nastiest agents in the world, with this delivery molecule, this cell penetration enhancer that can allow, when you inject it into the tumor, that the drug will disperse through the tumor based on the volume of the tumor. We set the dose based on the volume of the tumor, so you can saturate the entire tumor. It took us a long time in the clinic to understand exactly what the dose should be. But now, for the first time, you can inject a water-based system. This is a water product. And tumors which are consistent of high density, high fat, high pressure, and lots of stroma, but water and fat, water loving and fat loving. Now you have a product that is water loving and fat loving, but it's only water. And now this product will move through the tumor. It will kill the tumor. And as it dies, it stimulates a very potent patient-specific immune response. So for the first time, we've enabled an intratumoral drug made of water to be absorbed by a tumor made primarily of fat and water and stroma and dense tissue. And this is what happens. If you go on our website, you can see a number of places where we've injected into these tumors. They die. We can kill over 95% of these tumors. In fact, we can kill almost 100 of them, 100% when we get the right dose per tumor size. That leads to a systemic immune response. And so in our clinical studies, we've dosed over 200 patients in our clinical studies. What do we see? Metastatic patients, the tumors dying leads to the more that we can treat, the more tumors we can treat, the more drug we can give into the tumors, the longer the patients appear to live. And in a local setting, because obviously you have a local killing power, in breast cancer for women who were waiting for surgery, we did a study and we showed that we could kill greater than 95% of the tumor prior to surgery and stimulate a strong immunoactive immune activation, ignite an immune activation against the cancer. So we are killing in the metastatic setting whatever we can see. It doesn't have to be. You don't even have to bother with the small tumors. The immune cells will go after them, keep them in check, or regress them. And in the local disease, we kill up front, and then you add the chemotherapy to see if we can increase the number of women who would have in breast cancer what's called a pathological complete response. That means a dead tumor at the time of surgery. So what we're doing is 2 studies now, a phase 3 study, last date, in metastatic sarcoma, a deadly disease for which there is very few good options for patients, especially after they failed their first chemotherapy regimen. And another study in phase 2 in breast cancer for women who go through this brutal regimen of chemotherapy and in the hopes of trying to kill all the cancer. Probably less than half of the women in the population we've identified will have that benefit, but they can die from the chemo. And now what we're going to be able to do is hopefully kill the tumors, give the women a complete pathological response, and hopefully prevent the disease from coming back for a long period of time with immune activation. That's what we're doing locally. In metastatic disease, the phase 3 is in sarcoma. It's our drug, local therapy, head-to-head against the best systemic chemotherapy, and the endpoint is overall survival. So we have 2 big studies. We're excited about it. They're going to be enrolling patients. Well, they're enrolling patients now. I think we've seen some very favorable results, and we're looking forward to continuing these big trials and making this concept that I founded in my basement, your classic story of a guy with an idea sitting in his basement, thinking about something from a different perspective. I'm not a cancer scientist. I'm an engineer. And we've, 12 years later, after 200 patients, we're in the last stages of development. That's kind of the story we're telling.
That's such a fantastic story. And I really love when, when I see people from different backgrounds, like how you mentioned you have an engineering background, and then really bringing those diverse perspectives and looking at problems from different angles that perhaps, like you said, a cancer scientist, going by the books and having been in that box for years and years can't see. And so bringing that fresh perspective and those innovative ideas, it's really key. And it's a testament to really how multifaceted, the field really is and how complex cancer is. And so you do need, a plethora of angles and elements to really tackle it.
Well, also the concept of intratumoral delivery, injecting right into the tumor, has been around as long as there's been chemotherapy, 50, 60 years, because you want to localize it. The problem that I saw it as a chemistry problem. The tumors are dense, fat, stromal. The drugs are water. Water and fat don't mix. I had a technology that we created that I worked with for oral delivery. I thought it might be relevant. It was not obvious that it would work at all. In fact, we had to do a lot of experiments to find what was necessary to make it work. And now we can see that we can kill tumors. For the first time, you can take a needle and inject a very large tumor. Much bigger than ablation tumors. Ablation tumors, where you clear them, are maybe 2, 3, 4 centimeters. We can kill 18-centimeter tumors over the course of our that is a big tumor. We can kill it. We can convert it as it's dying into an immune activating site for the body to now key off of, go back to the lymphs and the immune activating areas in the thymus and the spleen, and stimulate a very potent anti-cancer immune activation that does shrink tumors that we didn't inject, and also battles the ones that may not be shrinking. And so it's very exciting. It's very fun to see a dream, because when I had this idea, I said, how could I not do it? Even though I was comfortable in a high-paying job, I thought to myself, well, if I really do have a highly effective treatment for cancer and I never did anything about it, how would that be? And so, almost 13 years later, it's been very gratifying. I have met the patients that I have helped or that the drug has helped. And I didn't help them. The doctors did. I gave them a tool. They used the tool. And it's very satisfying. I think every CEO should meet the patients that their products are helping or trying to help, get to know what they go through. I have spoken to many cancer patients. My family has had cancer. My grandmother died of cancer. My sister's had cancer. My breast cancer. My sister-in-laws have had breast cancer. These are, lives, and it's not just numbers. Every patient has a family. Every family cares for their loved one and they want them to get better. Now, we have a new weapon. This is a new way to kill. It is not like radiation or ablation, local therapies. This is a local new way that stimulates a global immune response. Patients feel good because the drug is harming the tumor and not them, and they live longer. And at least it looks like that is the case in metastatic disease, and we will hopefully prove it in the study we've got going. And then in the local setting, a lot of times, these women, as I said, they go through a harsh regimen, and to what end if it doesn't really get what they need? Now we have something that can add to the current regimens, and it's very, very satisfying to see the work that I've done actually result in a real good for the people that we're helping. I put friends into the study because I believe in it. What
Absolutely.
we're
Yeah, and I think just having the patient at the center, we talk about patient-centric trials and patient-centric drug development, but what does that really even mean, for you to have gone out and actually have met the patients, getting their feedback and then taking that full circle and then going back to them and seeing the results of your efforts. I think that's really testament
also
to everything. Yeah.
it also centers you as a leader. Whenever I am asked, to make a decision about something, I say, what is in the best interest of the patients? How will whatever I decide benefit the patients? Because, I think the chairman, the guy who founded Merck said, if you take care of patients, the business will be okay. And so whenever, people say, well, what do you want from this? You want a partnership, you want this, you want to sell it, you want to develop the drug yourself. I said, whatever is going to get this drug to the market fastest and benefit the most patients as fast as we can is what I'm going to do. That may be a whole bunch of different business things, but everything that we do, whenever I'm making a decision, I tell my people, let's step back. What's in the best interest of the patients here? Uh, yeah, look, it's always about the patients and focusing on the patients and making sure that the patients have your thoughts behind them.
Indeed, and I think that's. I guess would it be safe to say that that's one of the keys to success for you at Intensity, really to have that mindset.
It has been what we drive by, but also another thing is to be authentic about, and realistic about what you can and cannot do with the funds. We're a very small company. We're not funded by, your traditional large VC firms that incubate a company in their own mind or whatever they do. They find a technology out of a government or out of a high academic university. They put together business people, marketing people, development people, but they're not the people who originally, they're really putting together a sort of like a movie. You're putting together all the groups and everything. This is from a guy with an idea, in the classic sense. I study a lot of the history of science and the history of entrepreneurs. Wright brothers, George Eastman, the Kodak camera, Steve Jobs, and people who've really created value. And what you really want is that passion of an entrepreneur to drive forward and sacrifice what you need to do with the motivation of the patient behind you. The passion of an entrepreneur and the motivation of helping patients, I think it is very powerful, and it's gotten us, through a lot of very thin times. Markets are very challenging for biotech right now. We just raised $3 million. Grateful for the investors that came in. And all the investors, I have a relationship with all the investors, and they know the authentic nature of what we do comes out. When I talk to the investors, I'm telling them what I believe, and I'm honest as best I can, project that to the investors and the investment community. But ultimately, if you really are dedicated to helping patients, I think the world wants to help patients. And that's what I've found. I found there's been so much help that I've had from my existing investors, from the markets, from the partners we've had at the National Cancer Institute, at the OICR, at the Swiss SAC group, even at Merck and Bristol, where we partnered with them. I find that everybody wants to help patients. And if you're dedicated to that, everything seems to work out.
That's wonderful. Everything falls into place indeed. And so I think you touched upon the regulatory end of things, or perhaps not, but how do you see Intensity taking this product, and how do you see the regulatory landscape now compared to, let's say, 10, 20 years ago? There are a lot of different pathways, accelerated approval pathways, and then you have the oncology division specifically for oncology drugs. And so what are you looking towards when you're ready to get this product moving towards approval and commercialization?
Well, first of all, I have only had positive experiences working with the US FDA and the other regulatory agencies, Health Canada, and now recently the European Medicines Agency. I find the FDA, they want to make sure that drugs are safe and effective. That's their motive. A lot of times people don't care about that. They want to just get something where they can make money. But I'm on the side of the FDA. I believe we have to prove safety and we have to prove efficacy. And if we do it, and if we are intent about doing it and about doing it the way they would like you to do it, which is really a high level of science. I mean, I find the FDA to be just really good at the science, not only the regulations, but the science, and the guidance that they've given us over the years has been actually invaluable to proving what we do. I think it's a well-run organization, the FDA. I found the EMA to be very, very good. Our experience this past year has been, they have their questions, you answer the questions, you have the data to support your answers, they give you the go-ahead to start the study. So Health Canada, we're now, with the sarcoma study, we have authorization from the US FDA, the Canadian Health Canada, as you know, Health Canada, very nice organization. I enjoyed working with their medical people very much. Then we have the European Medicines Agencies. We're approved to start the study in France, Germany, Spain, Poland, and Italy. So those are the countries we're going in, and we've just recently gotten the Australian equivalent of the FDA to allow us to start the study. So I found that working with regulatory agencies are to be highly positive, very, very friendly. They want patients to do well too, but they want you to prove to them that you do have something that works and safe. We've gotten orphan drug status on our sarcoma. We've gotten fast track status in our breast cancer program for metastatic disease. We will intend to get further regulatory accelerated approvals, is what we're looking for with our study in breast cancer now. So again, if you follow the regs, if you give them good quality data and science, if you can communicate it, they will work with you and you can be very successful. That's been my experience, and that's been, I think, what's made us successful is focus on the science, just the data, show what it is, understand it, lead to the next set of data by understanding what you have. Because it really is a new way. We had to figure out a lot of things with this technology, and we've done it, and they bought into it, and our phase 3s were approved for start. What else can I ask?
Yeah, wonderful. You make it sound so simple. I love it.
Yes. Well, behind that simplicity is a lot of work. We are a small
Absolutely,
company,
yeah.
and everybody does a lot of things. And Yeah. So you have a phase 3 company that's been there for 12, started 12 years ago. We're in phase 3 with a very, very small team of people, but we're focused. If you're focused and if you go 2 steps forward and no steps back, that's a lot better than 2 steps forward and 1 step back. So by keeping the communication lines direct to me being involved where I need to be, we can focus on just what we do, which is to get these 2 studies done. We don't have a lot of other, we have one product, 2 studies. If either of those 2 studies work, it will be a very important movement forward in the field of oncology. And that's what we do, focus on that.
Mm-hmm. Indeed. And I think you mentioned you're evaluating this therapeutic both as a monotherapy as well as in combination with other treatments as well, including standard chemotherapy. And so, how do you envision this to be more of a monotherapy, or I guess it depends on the indication, depends on the stage of disease?
Exactly. It depends on the indication. The the sarcoma, we did 30 patients in sarcoma, and we did our drug alone, and we did our drug with immunotherapy, and there was no added value with the immunotherapy. And our drug, compared to the standard of care, was the signals that we got looked way better than the standard of care based on historical data and the synthetic controls that we ran. So, and the safety is so much better. So, these drugs in the sarcoma field, eribulin, pazopanib, trabectedin, they're extraordinarily toxic. Pazopanib will cause an elongation of your QTC, and you can have, heart attack. Eribulin is a 21-day cycle. On day 1, you get a dose. On day 8, you get another dose, and then on day 9, you're vomiting for a week. I mean, terribly terrible. And then you start to recover, and you're on that drug on a second and third line until either you die, until your cancer progresses, or just you can't take it anymore. That's not the way we should be treating patients. With our drug, we inject the tumors, as many tumors as you can, wherever you can get a needle to safely. The tumors die. We believe we're seeing a lot of that on the scans. The tumors become necrotic and cystic. And the immune system can get engaged. We've seen influxes into the tumors of immune cells. We've seen systemic increases in T cell repertoire and a whole bunch of other markers of immune activation systemically. And what we're seeing is the result of a drug that doesn't harm the healthy tissue. You don't have these grade 4, grade 5 adverse events, and grade 5 is death, and grade 4 is severe debilitation. You have 1 out of 9 only having a grade 3, which requires a little bit of hospitalization. Most of the adverse events are low grade pain because it's a long needle into the tumors, or fatigue, which could be an immune activation symptom, or some slight nausea on the day of dosing, which could be due to some of the analgesic or anesthesia that it's given. So we feel we have a very, very safe product after 200 patients. We know how to use it. We know how to dose it. And if these studies show the efficacy, we think there will be a game-changing effect to the cancer. Now, that's what we have to prove. But a local therapy to treat metastatic disease is rather unique and different. And the doctors are enamored with trying this. And if they see the results in the phase 3s or the phase 2 that we saw in the prior studies, I think we're going to have a large endorsement from the existing doctors in our trial. So it's all about execution and focus, and making sure that you keep your motivations proper, patience, not with the C, but with the TS, and doing everything. Although you need
You
patience
need
with the
both,
C,
I would say, yeah. A bit of both,
patience,
a lot of, maybe a lot of both.
a lot of both, especially in this financing market. But look, if I can continue to execute, eventually, everybody will be on our side. Everybody wants to help patients. Uh, that's what I find, and I've been helped because the patients are focused. And I love talking to the patients. I like talking to people who've had cancer. I love talking to people who want to be in our studies. Or if they can't be in our studies for whatever reason, I try to help them with clinicaltrials.gov to find a study that can be suitable for their sarcoma or their breast cancer or whatever. I'm not a doctor, but I can tell you that after 12 years of doing this, I have a good understanding as to what is possible and not possible with current care. And I hope we can now do some things that are way beyond the possible of today with this new drug tomorrow.
Your engineering mind and perspective, I think, is really brought, a candidate that, we talk about targeted therapies and targeted medicines, and what does that really mean? Is it just targeting one specific protein? Is that enough? Often not. But really getting into the biomechanics as well and combining that with eliciting an immune response and all of those things. I think that's indeed the cutting edge of innovation, to be able to bring all of those elements together, the chemistry, the biophysical properties of the tumor microenvironment, which I studied in grad school. So I definitely know a lot of, a bit about that. And so just to bring and to address all of the elements together, I think that's very key. And I think that's why you're hitting the nail on the head.
Well, it's exactly right. Look, everything is physics to me. Everything is physics. So why didn't intratumoral work? Well, the miscibility of a water-based drug and a fatty, dense, high stromal microenvironment of a tumor were incompatible. We found a way to make them compatible. The toxic agents are very strong. If they get into the cancer cells, they will kill them. And that's what we're seeing across a whole host of cancers, not just the ones that we're in the phase 3 and phase 2 studies with. I mean, this has worked in our phase 1 in 20 different types of cancer. We have seen the ability to kill cancer like pancreatic and colon and bile duct. And not every tumor is going to be amenable for injection. So not every cancer may be treatable with this, but a whole host of them, a whole host of applications can come out of this really novel new way to kill tumors. It is unlike anything else out there. And we're excited to see the doctors and the patients getting more and more enthusiastic, and the regulatory agencies, and, eventually, the investors and the people in the communities where this is really going to be helpful will be really the most enthusiastic. We've worked with talked to a lot of the foundations that are, the groups that have tried to help research in cancer, SPAN for sarcoma, or the Chordoma Foundation, a whole bunch of other things. So everybody will be on your side when you have something that works. And that's our goal, is to get it to be proven to be working.
Indeed. And I find it interesting also, going back to the mechanism of action, where you're able to have, I guess, a cytotoxic effect, where you're able to elicit tumor kill, but then also activate the immune response. And so what is the mechanism, I think, I wanted to ask you about in terms of that tumor kill?
Well, it's excellent question. So the two drugs that we use have both dual tumor killing and immune activating mechanisms. So cisplatin is something that will bind to DNA, cause an apoptotic cell death. Well, apoptosis is immune activating. Cisplatin also, and these are all published by a number of papers on it, cisplatin will also stress the endoplasmic reticulum to cause the release of two proteins, calreticulin and high mobility box 1, to the surface of the cell. And those are now able to attract T cells through a toll-like 9 receptor mechanism. So the death is immunological as well as death, right? Now, vinblastine has a completely different mechanism of action. Vinblastine is a tubulin inhibitor. It'll shred the tubulin. It's a very potent old molecule, but it shreds tubulin. And when that happens, it stops the cell replication process. Doesn't matter what the cycle is. In addition, it will mature dendritic cells in the environment where there's concentrations of vinblastine. Well, where do you want the dendritic cells to get mature? In the tumor. So now this is in the tumor. It's got it starts to mature dendritic cells. Now they can get a read. If this is the protein surface of the tumor, the dendritic cells can come in and start to read that fingerprint, and they go back and they train the CD4, CD8 T cells to come in and attack the cancer, not only that we injected, but what we didn't. So both drugs, and I did not know this a priori. When we started the screening, we screened about 8 or 10 different mechanistic drugs, cisplatin, a bunch of other ones. We looked at a whole bunch, and the top two were cisplatin and vinblastine. So I thought, okay, well, let's try them together. And it was 1 + 1 = 3. And we got a better both kill and survival and immune activation and tumor shrinkage in the mice. And it was all just step by step, systematically going through a process of identifying the best components, the best ratio, the best relationship for these three components of our chemical that is the cell penetration and dispersion enhancer. The two drugs came out to be 1 and 2. Let's try 1 and 2 together. That made a bigger number. And lo and behold, what we're seeing is very, very favorable safety uh, and very exciting early results that show efficacy could be extremely good in at least the two cancers.
That's so exciting where you're taking existing drugs and you're able to improve them by improving the targeting and delivery of them, which is amazing. And so I can imagine that this could be a plug and play system where you can, if there are new drugs that come out, you can plug them perhaps into this therapy.
Not only new drugs that can come into this, but also old drugs that were just too toxic to be developed because they were so potent, but they were going over the whole body and they were toxic. So this platform could revive really potent killing agents. I mean, there's a whole universe of opportunity. Yeah. So you can go back and look at, well, that's worked really well, but it was too toxic. Well, now if we inject it into the tumor and it's got immune activating and killing mechanisms that are better than the two that we've chosen, we can get next generation products. It's a matter of finding it. So it's a new way. It's going to open up a whole new world. I mean, I look at it as it could be a Wright brothers like moment. Now, these are two bicycle guys who decided that let's strap an engine to some fixed wing that they had to figure out how to make it and what it was and how to get most lift. They developed all the math equations. If you go to Kitty Hawk, to the Wright Brothers Museum, you get this wonderful exposition about how guys outside of the flying, heavier than aircraft flying field created, what they created, which has led to, enormous, since 1903, enormous change in our whole world. I mean, what was the world like before they had the Wright brothers planes, or Henry Ford and Otto Benz created the motor or the engine that could be powered by gasoline. It's all kind of builds into it. And now we have this delivery technology that I helped to create for oral ribelsis, now being used potentially as a way to treat cancer. Everything can lead to something else as long as you have what I call HI, not AI.
Human
HI,
intelligence,
human
intelligence. That's not going
yeah,
anywhere, hopefully.
Not going anywhere. The machines aren't going to replace us anytime soon in my mind. But ultimately, the innovation has to come from somebody thinking about something in a completely different way.
Absolutely.
With the experience to make it happen.
Exactly.
Not only know about it.
So what can we expect from Intensity in the coming months and the coming years? And what specific milestones are you looking forward to hitting?
So we're enrolling now in our phase 2 breast cancer study. We hope to have the data on pathological complete response rates between our drug with chemotherapy versus chemotherapy alone towards the end of next year. We're also enrolling in a 333 patient study in metastatic sarcoma. So the breast cancer study is about 50, 60 patients. The sarcoma is 333, as I said. So we hope to complete enrollment towards the end of next year. The end of next year would be great. And we'll know over the next couple of months. Both studies are open label. If you're getting our drug. It's randomized controlled, so the doctors can't play a game and say, I'm going to give this patient Intensity's drug and this patient the standard of care. But what they will do is once they have identified the patient, they will go in, they'll get randomized to our drug or alone in the sarcoma case. And so you can expect enrollment reported numbers. You can expect how many sites are being activated. You can expect potentially some insight as to how many people are alive or dead, depending on what information we get, how the safety is working over the next 2 years. Those kinds of things are what we're looking for. And then we'll have our endpoints on the phase 2. We'll have our endpoint in the phase 3. Probably the phase 3 endpoint is in 2026 at some time. We are taking interim looks, statistically significant interim looks at 20, 40, 60% of events. The endpoint is 80% of events. In the sarcoma study, the events of death. So it's a little bit longer. But if the drug is keeping people alive a lot longer than the standard of care, we should see that differential of staying alive and not staying alive meet sometime in 2026. So we'll have those data all throughout the next 2 years. And hopefully this study sarcoma study will result in a longer, healthier life for the sarcoma patients who so desperately need it. And women who go through this brutal chemotherapy with the larger tumors will be able to achieve an endpoint that is pathological complete response, a dead tumor, which we know leads to an event-free survival benefit. So those are the data we're looking for in the next 2 years, and it's 2 years will be here before you know it.
Absolutely, for sure. Those are wonderful, wonderful milestones and goals to have. I think you always mention the patient, which is so important, right? This is why we're having this conversation and why you are where you are doing what you're doing at Intensity. So Louis, thank you so much for being on the show and for your time to share all of the amazing work that's happening at Intensity.
Thank you, Ayesha. We could have another 10 hours of conversation.
All right, that's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we shall see you next week. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.